Good afternoon. I'm going to be talking about subjects that we've had previous lectures on by my colleague Joel Wertheim on the origins of coronavirus, and I in November spoke to this group about coronaviruses as well. And so I'm not going to be covering that topic very broadly, but focusing on a comparison of the HIV epidemic and the coronavirus epidemics and what they may teach us, and more broadly, how they may help us manage other impending problems that we know are growing more serious for us. We thought that these epidemics are worth thinking about it because of their huge medical, social, and economic consequences. Like climate change, they result from human overpopulation and encroachment on remote ecosystems to hunt wild animals. They provide lessons for control of future pandemics and more broadly for control of other unpredictable natural disasters, such as earthquakes, volcanoes, and the effects of climate change, including sea level rise, ocean acidification, droughts, and extreme weather events. So that's may sound a little grandiose, but I'm hoping that maybe there are some lessons for us for many of these disasters that I think keep some of us up at night. We will look at the benefits from the immediate and appropriate scientific, medical, and political actions to control both the epidemics and the consequences for the course of them from ignoring, politicizing, or allowing the spread of misinformation. So these are both zoonotic infections, which are transmitted regularly between animals and humans by a variety of routes. Many previous pandemics have resulted from endemic bacterial infections that are infrequently now transmitted to humans, such as plague. This is a bacteria that's transmitted from rodents, mainly rats, via fleas, and has caused multiple epidemics beginning in Europe in the 1300s, with really massive consequences on the development of Europe over this period. About 800 cases currently occur worldwide annually, and the organism that causes it, Yersinia pestis, is endemic in California in the ground squirrels, particularly up in the Sierras, and they do lead to rare cases a decade here in California. Endemic viruses such as influenza A is maintained generally by human-to-human airborne spread, causing winter epidemics worldwide and oscillating between the northern and the southern hemispheres. There's a large reservoir in ducks, chickens, pigs, and other animals, and dramatic pandemics, such as the one in 1918 and 20, have caused massive disruption and death. A current epidemic in domestic birds has necessitated slaughtering millions of birds to prevent spread and could pass to us. Newly emerging viruses that we want to concentrate on today, however, have just moved in from animal reservoirs to humans, and we'll address that in more detail in a moment. So HIV originated in remote areas of Central Africa, thought to be the Cameroon, between 1900 and 1930. This has been deduced from looking at the phylogenetics of current strains and calculating back to how long ago the first strain in humans would have had to have evolved. in order to account for the changes that have occurred in the last roughly 90 to 100 years. It seems to have arisen from hunting not only chimpanzees, which has caused the HIV strain that currently causes the worldwide epidemic, but also smaller numbers of cases have been documented from gorilla viruses and from sooty mangabe viruses. So there have been multiple simian immunodeficiency viruses that are common throughout all the primates to humans. Genetically, similarity of human genes to those of chimps, the host genes, that is, about 98% or more are shared, and our similarity to primates like the gorillas and so on, have enabled these SIVs to adapt to humans when they ended up in human bodies. Why would a virus that's been presumably in these primates have only first been found in the 1980s in humans? This event, we think, occurred in small, isolated rural populations. And as we've calculated, perhaps it took 30 to 50 years for them to be recognized, in part because there are no medical services or experts to recognize new diseases in these populations. Also contributing was the rise of cities in the Congo Basin in the 1960s that allowed amplification of HIV primarily by heterosexual sex in the newly forming urban populations. The Belgians ran the Congo very poorly for many, many years and educated no technical expertise and so on in the indigenous population. As a result, when they left, there was a big problem. And we'll talk about that and how that evolved. Viruses that in this case came from bushmeat, and you see a professional hunter there with his harvest of dead primates in the sack on his back there, we think have jumped species many times, but only really had a global impact with this transferal around the 1920s. Many viruses, when they first jump to humans or to other species, perhaps, we know less about that, become more virulent and deadly. And this is really counterproductive for them because it increases, and because this increase in virulence kills the human host on which the virus is totally dependent to reproduce. So it's like burning your house down while you're in it for the viruses to be deadly. Many viruses begin to evolve to a less damaging state with their or equilibrium with their hosts, allowing them to spread more efficiently because the hosts don't die before they can transmit it. Now, one of the questions is how did chimps and when did chimps get the SIV chimpanzee that we think was the origination of HIV? And we know now from examining the genome carefully of many primate species that two primates appeared to have contributed. On the lower right, you see the red-capped mangabe and the greater spot-nosed monkey, and their SIVs appeared to have infected a single chimp that allowed the recombination of the genetics of those two simian immunodeficiency viruses. And it's been estimated, again, by my colleague Joel Wertheim to have occurred in the late 1400s with fairly wide uncertainty around that. We know that chimps hunt and eat other primates, and we think that because of that, a single chimp could easily have been infected with 2 SIVs, and this enabled their recombination. One of the questions about this transfer was it immediately raised the question of whether the wild chimpanzees themselves are in any way affected by this, and how common it is among chimpanzees and so on. And so in what I consider rather heroic field studies, a number of virologists tramped into the jungles of West Africa and elsewhere, and obtained samples of urine and feces in which the genetic sequences of SIV could be found. And they found quite a lot of variation from 0 to 35% rate of positives in various groups of chimps. They then began to study a unique group of chimps that have been observed by Jane Goodall and her colleagues at the Gombe Stream National Park in Tanzania, and where they have the ability to almost daily keep an eye on these animals, and they know them personally. So they know and can identify, they have names for them and so on. And so they can track by collecting stool and urine which ones are infected, and then see what happens to them over a period of time, a kind of classic longitudinal study. And so a couple of things have emerged from that. First, that there was some variation in the prevalence, that is, the percentage of the population that was infected in these Gombe chimps. In 2002, it was 9%, in 06, 18%, and in 08, 14%. And it's not clear whether that's an increasing curve or whether that's simply variation. They could tell that there was both sexual and maternal transmission to newborns, and that the outcome of SIV was a considerable increase in mortality in the infected animals. 41%, or 7 of 17 infected SIV-infected animals versus 14%, 11 of 77 in the negatives. And that's a 16-fold higher age-specific mortality when you adjust it for the mortality you expect at age from other things and so on. The fertility rate was also affected with a 3 times increase in infertility. And it wasn't clear whether there was gestational and infant mortality, that is the death of the fetuses and the newborns from this, but presumably there was some transmission, and it's very possible that this is unrecognized and unmeasured mortality at this point. Now, since we've established now that HIV originated in Africa, how did it get out of that? And it turns out that, again, the Congo is the focus. In the 1950s and 60s, it seems to have spread from rural to urban areas in the Congo, particularly Leopoldville and Brazzaville. And in 1966, it then was brought to Haiti. And this was, again, the consequence of the poor management of and transfer of the Congo to its inhabitants by the Belgians. This necessitated that the new governments hire as contract workers professionals of various kinds, doctors, nurses, lawyers, engineers, accountants, all sorts of people. And they realized that Haiti was a potential source of such people, since it's a Francophone country, as is the Belgian Congo. In the late 1970s, an epidemic strain then found in Haiti was introduced to the US, possibly by gay men who visited Haiti for sex. That was a well-established phenomenon. And by 1978, in retrospect, once we got the ability to measure antibody to HIV in gay men, we were able to document that going back to a hepatitis B study in San Francisco, 4% of the gay men there were positive in 1978. By 1980, the first cases were recognized and publicized. One of the questions then is, why did it spread so slowly initially and then widely and effectively? And as I've indicated, we think it smoldered in remote, inaccessible areas in the Cameroon. And then as trade and transportation was increasing, was moved to these larger cities where it could be amplified. And so it certainly spread widely and effectively for a number of good reasons. It has the virologic characteristic of having many genes that control its replication and allow it to smolder within an individual for an average of about 10 years before symptoms of AIDS appear. Now, that's just an average. Some people get sick within a year or so of infection, others a lot longer. This means that HIV-infected persons remain well and sexually active, but infectious to others for long periods of time, and certainly enhances its transmissibility. The urbanization of the Congo in the mid-20th century created, as I say, the right socioeconomic and geographical factors needed to transfer this to places that it could then leave the continent. And gay men were increasingly sexually active, and there were widely traveled homosexual men. And I'm going to show you an example of an airline steward who used gay bars and baths for anonymous sex with multiple partners. This is a graphic of an investigation done early in the epidemic by the CDC to try to understand the linkage among a number of cases, starting with patient 0 in the center of this cluster. And the colors here indicate two of the most common and easily recognized manifestations of HIV, because they are uncommon otherwise and were very common in the HIV patients. And this Canadian air steward was flying around, and as you can see in the left side of the first ring coming out of from him on the spokes, he infected a number of people in LA, and on the right side, a number of people in New York. They then generated secondary cases in those areas, one in Florida and another chain in LA and so on. From New York, it created a whole series in the lower right-hand corner of this in the New York area. So it was first recognized in Los Angeles, and this is a report from the CDC's weekly report on morbidity and mortality and basically infectious epidemiology, in which 5 young homosexual men who had been cared for at UCLA had the combination of pneumocystis pneumonia, now called pneumocystis jirovecii pneumonia, and cytomegalovirus infections, a herpes virus infection. And importantly, they had no known immunosuppressive drugs or diseases that could explain this. And they, for really fortunate reasons, UCLA had just learned and developed the technique for measuring a subset of human lymphocytes, the CD4 lymphocytes in blood, that they knew were important to immune responses and immunology. And they found that these were profoundly depleted in these patients. So that put together the infection and perhaps the mechanism, the loss of a major component of the immune system. The initial reactions to this and the developing recognition throughout the United States in the major cities where gay people tended to congregate of a new problem. And the initial assumptions about the disease were that it was limited to sexually active gay men, that small numbers of persons were at risk, that it would be self-limited like our previous experience in 1976 with the Legionnaire's pneumonia outbreak among veterans in Philadelphia. And those of you who are old enough to remember that will recognize that that was a bacterial infection caused by inhalation of these bacteria from the condensers of air conditioners in the hotel that hosted this convention. And so that outbreak, if you will, rapidly resolved. And it was, while it was quite deadly to a number of these veterans, it was not something that spread widely. We've since recognized that we do have this throughout the country and that it is often in that setting of downwind, basically from air conditioners. There was fear of contagion in the healthcare facilities, leading to physicians and paramedical staff refusing services and hospitals directing patients to academic centers with more experience, but mainly because they didn't want any part of this because they were afraid it would scare other patients from coming to their hospitals. There was extreme avoidance, really, of it in many segments of the population, except for several groups that began to focus and realize what the potential for this was. Epidemiologists who had been focused on sexually transmitted diseases at the CDC led the epidemiological investigations, and small numbers of cancer and infectious disease physicians at urban medical centers started being willing to see such cases, as I was with Chris Matthews here in UCSD. And then we began to realize we had increasing caseloads. And I'll explain a little later, we really were set up to notice this quite well because we'd recently started a clinic for gay men trying to deal with the large problem of other STDs or other sexually transmitted infections in them. You might wonder why cancer patients, but of course the Kaposi sarcoma is a cancer, or at least simulates one very well, and was then under their purview. As the epidemic evolved, we began to recognize risk groups, and I kept going on local television and explaining why maybe IV drug users and transfusion recipients, Haitians, and then infants and children and so on might be infected, and how. And our colleagues in Europe noticed that wealthy Africans began coming in with this syndrome, directing our attention really for the first time in my experience, to the African origin of this. Thus, we learned that HIV was transmissible by homosexual and heterosexual intercourse, blood from transfusion or needles, that is, contaminating needles that IV drug users might use, and perinatally during childbirth and during breastfeeding. There were some fetuses that were infected and tended to lead to miscarriage. We knew fairly quickly that HIV had a prolonged asymptomatic period, I mentioned before, produced a high prevalence of generalized lymphadenopathy or swollen lymph glands in the region of the neck, under the arms, and in the groin, and so on. And at that point, we were following in our clinic about 100 San Diegans, and found that about half of them had this this sign, and we presumed and proved ourselves subsequently correct when antibody testing became available, that they in fact were already infected. We didn't know whether they were going to progress, but we knew at that point that the AIDS cases we were seeing were a tip of the iceberg phenomena, if you will, for HIV in the gay population. Now, HIV was around for several years before we were able to identify the cause, and we learned about it from the French in the laboratory of Luc Montagnier at the Pasteur Institute in Paris in 1983 and 4. He had isolated from the lymph node of a French AIDS patient, a retrovirus that was distantly related to one that Robert Gallo had previously found in patients with lymphomas. And he and his colleagues were subsequently recognized as the discoverers of this with the Nobel Prize in 2008. The discovery of HIV enabled the development quickly of diagnostic tests, of antiretroviral drugs. As you can see, it took a year or two to do each of those, but we then had a series of drugs that we mistakenly thought we could use one at a time, and eventually learned that we had to give 3 at a time and involve new classes of drugs. Before, in 1996, we really were able to completely suppress HIV replication in our patients. And importantly, we rendered them unable to transmit HIV. And that was just a wonderful and enormous development. Meanwhile, we were hoping for a vaccine, but that has been pursued for 35 years with no success, and is completely understandable given the highly mutable genome of this virus. So HIV was a perfect storm pathogen in many ways. It had a long asymptomatic period. It was highly mutable, and it was latent in cells in a way that made it difficult for any antiviral chemotherapy to cure the patients. And so antiviral therapy had to be lifelong. And we knew that if people were careless and generated resistant mutants, that these would reemerge if we stopped the ART, and that it was a irreversible pathway if you sort of stopped your drugs inappropriately and allowed these resistant mutants to develop. We also recognized that there were social features, and we've mentioned a number of them, that sex behaviors are repetitive and provide multiple opportunities for transmission. And it turned out when you did the calculations, there was really low risk per episode of intercourse, but repetitive contacts with infected people would eventually lead to infection in a large proportion of the people. Stigmatizing or stigmatization initially was not led to it not even being acknowledged or discussed by our political leaders, noticeably President Reagan, and it didn't become a social or political priority. The situations in which transmission could occur tended to be poverty leading to transactional sex, the absence of legal or economic power for women within marriages made it difficult for them to refuse sex to their husbands if they recognize that they might be infected, and rape. In addition, HIV denialism and conspiracy theories based on antiretroviral therapy led to hesitancy and problems with that I'll show you below. After it was proven to be a cause of AIDS, a few scientists, including several from California, began a campaign to discredit the concept that HIV caused AIDS. Among them was Peter Duesberg, a professor of virology at UC Berkeley, and Carrie Mullis, a distinguished biochemist who was also an avid surfer, an alcoholic, I can attest to, and a La Jolla resident. He won the Nobel Prize in Medicine for inventing PCR, the polymerase chain reaction, a absolutely revolutionary method in biology for making millions of identical copies of DNA from a very small number. And I mentioned that I could attest to Carrie's alcoholism, not because we went to the same AA meetings or anything, but only because I attended a party where he was very raucous and out of control. The consequence for this, the important one, in some ways, or most important one of the denialism, was that the president of the Republic of South Africa, Thabo Mbeki, adopted this view that the disease was really one of social factors. He was a Marxist, and he thought that things like poverty and malnutrition, poor hygiene, sexual excess caused the disease, but not the virus. And that was the basic alternative hypothesis that these scientists had adopted. And Mbeki found that on the internet and thought, oh, that makes sense to me. So he did not allow any of the new drugs into his country for many years, leading to an estimated excess of about 300,000 deaths from AIDS in the South African population. So now that we've sketched out HIV, let's talk about COVID in similar terms. So we've had a series of epidemics of influenza A and coronavirus, the two major respiratory viral epidemics over the last 100 years, roughly, beginning with the Spanish influenza epidemic that affected 50 million people back in 1918, the Asian influenza epidemic that infected 1.2 million people in 1957, and may have been my first encounter with influenza. By 1968, I had gone to medical school and was an intern in medicine, and the Hong Kong flu epidemic occurred, again, infecting at least 1 million people. And I was pretty exhausted trying to take care of all the patients that poured into the Vanderbilt Medical Center as a result of that. Now, I didn't take care of all of them, but a lot of people did. We then had a gap of some years before the SARS epidemic, and coronaviruses began in 2002, small numbers involved, followed by a new influenza epidemic in 2009. And finally, the Middle Eastern respiratory syndrome, the MERS syndrome, again, a small number of patients in 2012. And then finally, with culminating in the COVID epidemic of over 660 million people and counting. So I've kind of sketched out here the comparison of these three coronavirus epidemics, and probably should have made a table to make it easier for you, but underlined is that in the SARS epidemic, there were over 8,000 cases now, 774 deaths, which is about a 9% mortality. In the Middle Eastern respiratory syndrome epidemic in 2012, there were greater than 2,500 cases, 866 deaths, and that's about a 34% mortality. So we might have expected that the COVID epidemic would be somewhere in that very high range of mortality. Fortunately, that hasn't been the case. The coronavirus epidemic now at 755 million confirmed cases, and no doubt many, many more, has led to 6.8 million deaths, and has a mortality thus of only about 1.2%. And as I mentioned, the global estimation we know is a major underestimate because people simply aren't counting, including the Chinese over the last several months, since they have dropped all their controls and have pretty much allowed the virus to race through their population without any real attempt to measure what's happening. Now, the origins of these coronaviruses that have caused these three epidemics are summarized at the bottom here fairly efficiently, I hope, that SARS-CoV-1 is endemic in bats in China and Southeast Asia. It seemed to have spread to civet cats and then to a Guangdong wet market where live cats were sold and housed. And that seems to have set off that relatively small epidemic in 2002. MERS appears to have been transmitted from bats to camels and from them to humans. And the SARS-CoV-2 epidemic we'll discuss in more detail here, but it seems to have originated in bats. And then we don't know for sure the mammalian host that would have been housed in the wet market in Wuhan, where it was first we think, transmitted to people. You can see a pangolin here pictured. This is a mammal and not a reptile, even though it looks like one. And they are widely and illegally trafficked across the world because they're easy to transport. They curl up in a ball and they don't need to be fed and so on. And but they're also in China a food source. Now, going back to the SARS-CoV-1, we know that its ancestor was produced by recombination of bats, recombination in bats in Southeast Asia, and was then transmitted to civets and other mammals. These virus-infected civets, and you can see what they look like below, were transmitted to a market in Guangdong and spread among the civets in the market. And then the virus underwent further mutations before being transmitted to humans. That may have been through another mammalian host, we just don't know. And it was rapidly spread to airline passengers and then into Vietnam, into a hotel, and to North America and Toronto and so on. And it really, because it was so deadly, there was very effective and rapid control of it. For SARS-CoV-2, and the COVID-19 epidemic, the outbreak began, as nearly all of us know, in Wuhan, a city in central China. And it probably originated in wild animals housed at the Wuhan wet market. And that's based on the fact that captured or farmed wild animals have been slaughtered and sold for meat in Chinese markets for centuries. So this is a well-established pattern. Even though the Chinese government denied that there were such animals in that market at that time. Of course, the fact that we had a model for this in SARS-CoV-1 was also helpful. And the geography of the cases in the outbreak in the city of Wuhan was centered on this market. And then finally, the alternative hypothesis we'll visit next was not supported by studying the genetics of SARS-CoV-2 from the first cases to the coronaviruses studied in this nearby Wuhan Virology Institute, which I think many people thought had to be the source since it was the major research center, in China for coronaviruses. It's certainly a logical alternative source, and it's still undecided, but the genetics, as I say, did not match. The closest relative of SARS-CoV-2 has been found subsequent to the epidemic in bats in caves in the northern Laos near the Chinese border, certainly an area where they may have been captured and or infected the wild animals captured in that area. And there are 4 scenarios that have been proposed based on 2 issues with respect to how and perhaps why this epidemic could have come from the Wuhan Institute of Virology. The first is based on release to infected humans. Was it accidental or was it intentional? And I think most people think that it's very unlikely that it was intentional if it came from that institute. And there's also been an issue raised about genetic enhancement. Were the virologists there doing studies in which they engineered in the laboratory a virus to be more transmissible or deadly to humans? These are called gain of function, but of course they also could represent bioweapon research. There's, I think, I don't think there's a good reason to think that that was occurring, and certainly it's been denied by Tony Fauci and others. The Chinese government has really been pretty uncooperative and covered up the connections in the following ways. It firstly falsely denied the presence of wild animals in the market, and yet they had been documented by a scientist there in photographs taken 2 weeks before the epidemic started. They intensely sterilized the market building before it could be examined by outside sources. And so we think that that really could have nailed the evidence needed to make the market the source, although obviously a virus from the Institute could have gotten into the market first. It seems less likely. Also, both in China and the US, the origin has now become politicized, and we're currently hearings are planned in the House Intelligence Committee on this issue to examine, among other issues, if Tony Fauci committed perjury in testifying that the NIH funded none of these gain-of-function research projects at the WIV, and he confirmed, and they've been very open about the fact that they were supporting coronavirus research there, based on the fact that even the milder coronavirus infections are a cause of a major morbidity, loss of work, and that sort of thing, from colds and that sort of thing. Now, moving then to the genetic variants of SARS and CoV-2, 5 major variants have rapidly become more dominant in their regions of origin, and then spread worldwide to create this epidemic, or a series of waves in the epidemic. And they're listed there. And the one that we're currently dealing with in the Omicron variant originated in South Africa, was detected early there. And the South Africans did a good job of warning people and helping people get access to the genetic code and so on of a new virus that took over the disease in their country fairly quickly. So all these genetic mutants change the amino acids in the part of the spike proteins called the receptor binding domain that binds the virus, and it must bind to the surface of a cell in order to infect a cell. Its target receptor is the ACE protein inhibitor in humans and animal cells. And these changes have enabled it to spread faster and replace the original strains very quickly, and have reduced the effectiveness of COVID vaccines by making the new variants resistant to the antibodies produced by these vaccines, and perhaps also by other mechanisms. So here are here's the time of the timing of these various epidemics, ending over here with the Delta being the most dramatic, and then the Omicron, which we encountered this summer and are currently dealing with, and it continues to mutate and create new variants that we are not entirely sure will remain well behaved, but so far have. So you can see the Delta epidemic has really, throughout the world, and particularly in Europe and the Americas, where it's more easily identified, created the greatest problem. And we were quite concerned that the Omicron variant would reproduce that of the Delta agent, since it seems to be very highly transmissible, but fortunately it has not. The variants increase transmission to COVID and reduce the effectiveness of vaccines or of prior COVID infections. And they emerge because they can replicate faster. And in the case of Omicron, it's interesting. They replicate faster in the upper airways and tend to cause more severe symptoms there. I recently had COVID where the only symptom I had was a sore throat and was not otherwise very sick. And we believe this is because it's not as well adapted to going deep in the lungs, from which it doesn't have any great advantage. And so just infecting the upper airways where it can pass to the next person more easily and more efficiently seems to have been the way it went in terms of being selected for these mutations. And it appears to evade the immune response of people who've been infected before or vaccinated to some extent, but certainly not entirely. Why do variants, or what are the things that make variants more likely to occur? We think, of course, infection of large numbers of persons simply increases the likelihood of mutations that can occur. Also, we know that prolonged infection of immunosuppressed patients, such as people with HIV on chemotherapy, taking immunotherapy for transplants or for arthritic diseases or other diseases like psoriasis and so on, all tend to lengthen the period of time when people continue to carry the virus and continue to have the opportunity for mutations to occur. And then finally, there's no evidence for this, but the possibility is that we could ping pong back and forth between humans and animals. The animals may carry the virus longer and allow more mutations to accumulate, but that has not been supported so far by any evidence. So we are recognizing now that we are in a period of where our herd immunity has allowed us to become less restrictive in the way we interact with each other. And this is based on the concept, and it works for many diseases, that when enough of a population becomes immune through either vaccines or infection or both, it reduces, in this case, the risk of COVID to the rest of the population. This can be achieved by a combination of both infection and vaccine, even if the vaccine's not 100% effective by any means. And reducing the severity of illness reduces the burden and the health on the healthcare systems and on providers and economic and social effects. So it's very much to our advantage to get people vaccinated. And vaccination, of course, works if the vaccines are effective, and the mRNA vaccines that most of us have taken certainly are highly effective in the range of over 90%, and the coverage, that is the percent of the population that is vaccinated, and that, of course, has not been as good. So just to summarize the COVID vaccine and variants, we've got great vaccines that work primarily against severe disease and also decrease the important post-COVID syndromes, which I haven't addressed today, but may become and have been documented to be a real drag on the healthcare system and the economy that does not that persists in people for months to years. Local and systemic side effects of vaccines are common, but mild and short-lived. And serious adverse reactions are uncommon, but certainly in a few cases can be quite dangerous or deadly. The variants spread rapidly and can increase transmissibility and reduce protections from vaccines, as I've just reviewed for you. We still have problems with decreased, excuse me, vaccine efficacy in special populations, those who cannot respond to the vaccine, and the durability of vaccine-induced immunity is certainly not something we would prefer, like 5 or 10 years or whatever, and seems to be reduced measurably by 3 to 6 months after vaccination. And we know that we're going to have to keep pursuing these variants, and now, as you know, have mixed wild type and variant vaccines that sort of update the immune system on what's circulating now. And we've got these problems with vaccine hesitancy and poverty that threaten global vaccine uptake, which is certainly going to be necessary to control the pandemic, although just natural infection repeatedly may be what we'll have to settle for many people in the world. There are a lot of problems with effective global vaccination, geographical dispersion, political and religious diversity, and thus opinions about the appropriateness of vaccines, and limited resources. There are many people in the country who are unwilling to be vaccinated. 80% have had at least one vaccine, but vaccine hesitancy is more common in the older patients who are more likely to be adversely affected, in the less educated, and in politically conservative Americans. Vaccines are moderately expensive. They've been paid for by the government up till now in this country, but throughout the world, this is going to be an issue. They require refrigeration and sterile injection equipment and some medical training and skill. Although it may be that we're going to eventually evolve better vaccines that don't require so much infrastructure and so on. The waning of immunity over 4 to 6 months and the continuing generation of variants may require semi-annual or even more frequent boosting, although most experts are hoping we can get by with just annual boosting. And that's, I think, something to be seen, and will depend to some extent on what variants occur. And a lot of these things undermine access, long-term acceptance, and the effectiveness of vaccines. Now, COVID denialism and delusions and vaccine hesitancy arise, we think, in kind of two forms. One is the lie that the government designed this vaccine to justify controlling the population, perhaps by injecting some sort of monitor in patients, or that the vaccines are simply a plot to make money for vaccine companies. Another point of view is that the whole thing is less serious or widespread than the government and the media portray it. There are a number of false beliefs about it, that scientists created and released it, that injections contain devices to track us, that scientists and political and public health officials have exaggerated the seriousness for political reasons. Vaccines are untested and therefore dangerous, and all restrictions on indoor gatherings or requirements for masking and distancing are unnecessary, illegal, or dangerous. So just to finish up here, I just want to compare here some statistics for the impact of these two epidemics, starting with the cumulative number of cases. As you can see, COVID leads by almost tenfold. About 2/3 of a billion people have had COVID, and that may be an overly conservative, poorly informed estimate. A better estimate for HIV of about 77 million. Global cumulative deaths, on the other hand, HIV in its 45-year or whatever period of pandemic has accumulated 40 million deaths compared to under 7 in or about 1% of all those infected. In COVID, we have 37 million living cases of HIV, and they represent a considerable burden on healthcare systems throughout the world and government budgets and so on. New annual infections are much more common, obviously, in COVID, but much less consequential. 1.5 million in HIV and 136 million extrapolated from rates back in 22 and more recently. And the cumulative vaccine rate, which of course is only relevant to COVID is 81% for one shot and 70% for 2 or more shots. And it's estimated that about half the American population has been infected. That may be an underestimate. We should have better data on that, but don't. So I think I'll stop at this point since I've gone over time, and let's open it up for some questions.
Thank you so much. Yes, we have a question from Bob Knox.
Yeah, thanks, Alan. You mentioned in one of your slides that in many cases these viral things jump from bats in some of your illustrations to some other mammal and then eventually to human beings. I'm just wondering if there are instances among the wild creatures that the transmission of this from, say, bats to some mammal has had significant impacts in terms of either infection or death in those animal populations before it ever gets to human beings, just as a point of comparison.
Sorry, I'm back. I got cut off there.
Oh,
Bob, can you state the question again? Sorry to.
yeah, no, that's okay. I was noticing in one of your earlier slides that the path of transmission of these viruses, newly emergent or whatever, has often been from a bat population to an intermediate mammal population, and eventually to human beings, with all the impacts in terms of both cases and deaths in the human population that you just mentioned towards the end. I'm just wondering if there are any measured examples of the impact of one of these new viruses moving from bats, say, to the next intermediate mammal population that have had similarly outstanding impacts on that animal population in terms of either cases or deaths that's ever been observed. I don't know.
It's a very good question, and I haven't seen anything about that. That could be done experimentally in small numbers of mammals, certainly, but let me think. We think that in artificial circumstances like mink farms in Denmark, transmission from humans to the minks was very dangerous and killed some of them, but most of them had to be destroyed because they were thought to be a danger to humans. And I don't know actually what that mortality rate was in the minks, but certainly there are many other mammals that are susceptible to SARS-CoV-2. I don't know also how well the camels handle their infection with the MERS agent. Something I should know, but don't. And that is known, I'm sure.
Thank you.
A question from Richard Moore.
Hi, you mentioned this briefly, but I was wondering if you could help me understand better why it has been so difficult to develop a vaccine for HIV. Okay.
It's a matter of at the level of the molecular virology of the virus comes in as messenger or as RNA, and it needs to be translated back into transcribed, back into DNA and enter the nucleus of a human cell in order to complete its life cycle. And so there's this very critical enzyme, happens to be the target of a whole class of our antiretroviral drugs, called the reverse transcriptase. And reverse because in our bodies, DNA gets translated into RNA, messenger RNA, and so on. That's the pathway. In this case, we're starting with the RNA and going back to DNA. It then reverses in our cell and comes out again as RNA to make the new copies of the virus. That reverse transcriptase is very prone to making errors when it does this reverse transcription process. And so it makes lots of mutants. Most of them probably don't get reproduced in any way. And they're fatal, basically, to that particular strand of DNA. But a few of them, because so many are generated, gives the virus an advantage. Either it counteracts an immune attack that's occurring on the virus, or it counteracts the effect of a drug. And our colleague Doug Richmond here at UCSD contributed valuable evidence to both of those mechanisms. First, he identified that early on in the 1980s that HIV could become resistant to AZT, and that it did so by becoming resistant to this. It did so through this mechanism of genetic mutation. And then he showed, with respect to resistance to immunity, that as a patient is infected with a strain, he begins to make quite effective antibody that begins to kill that strain. But then the virus evolves away from that particular configuration that the antibody recognizes, and it then can go on, even though that antibody is present in high amounts in the patient's blood, it simply cannot touch a newly evolved virus. Now that was pretty complex and prolonged. I'm sorry for that, but you get the idea. I think that that it's just a highly mutable virus, and that's probably built into its structure in order to allow it to persist in people, or in any situation in SIV and other primates, in order to simply evade the immune system.
Thank you.
A question from Barbara Parker. If you could just unmute yourself.
Thank you, Alan, for that. It's a very nice review. We're still losing 400 or more patients a day due to COVID, or with COVID, or as COVID, with COVID and something else. I'm curious about what is the primary cause for these people dying? Are they dying from heart disease and COVID as a secondary cause, or is COVID still driving death rates?
Yeah, that's
how significantly is COVID still driving death rates? I'll put it that way, given that the Omicron variant is milder in general in the population.
Well, as you know, probably much better than I do, determining exactly the pathway to cause of death and so on is very difficult, something that we confront when we sign death certificates. And so the question is always, was this a necessary and sufficient cause? That might be the criteria you'd use for a COVID death. Or did it simply take somebody whose lungs were already in terrible shape and just push them that little bit more? And they probably, any insult to the lung could have done that, if you will. Mm-hmm. So it's a little less specific. But I have trouble with, there have been attempts to not count deaths in which there was any other possible cause. And I think that's ridiculous, because we know that if somebody has lung disease or heart disease or whatever, they certainly are going to die, maybe with that, maybe of that, but they may not have been pushed over at that particular time. And it's
so, as far as you know,
Go
Yeah,
ahead.
as far as you know, we're counting deaths the same way we did at the beginning of COVID 3 years ago.
I don't know that for sure. And
Okay.
I've not seen that well studied. And it's a difficult one. I think that in this country, we probably are not so afraid of making the diagnosis, but in places like China and so on, it's a big problem. And you may remember early in the epidemic, the physicians and so on who were trying to bring attention to this in Wuhan were very badly treated. One died of HIV, of COVID himself, and so on. And it's in certain societies where they don't want to hear the news, it's dangerous
Right.
to make that diagnosis.
Thank you.
Thank you.
And then the question from Stephen Adler.
Thanks, Vanya. Thank you, Alan. Lovely presentation. I appreciate hearing it. So this is perhaps a more local question, but I think it speaks to a national policy issue. To what do you attribute UCSD's or the states or the federal government's pulling the rug out from under the more aggressive testing and vaccinating programs that were in place for the last few years? You can't apparently get a vaccine at UCSD anymore or a booster under normal circumstances. We know they got rid of the sites, but even going through primary care, for example, you can't get a booster, and apparently they're being besieged by angry patients who do want them. But it speaks to a larger issue, and testing as well is very hard to get, as you know, now, other than the home tests. Is this because fish rots from the head, starting up at the level of the CDC and or the government, or is it more locally decided?
Well, it's a great question. I've wondered about it myself. It almost looks like the Chinese copied us in a sense, because they kind of just, they were completely trying to control everything, and then suddenly let loose of all the controls without, any kind of preparation. And I feel like we did the same thing, a little more gently and measure in a measured way, but why and so on is not entirely clear to me. There's certainly been a lot of undermining of the question of efficacy of masking, for instance, and so on. And I wonder if people in government simply said, this is kind of our good enough state that we're going to have to live with, and we can't let the economy or whatever and the education of the young and so on and so forth suffer as they have by continuing these more restrictive policies. But that's strictly a guess, and I just don't really understand what the decision-making process was. Disappointing, I'm sure. Steve, I wish I had a better answer for you.
Thank you. Do we have
Thanks, Alan.
other questions from our participants? I'm not seeing any. So yes, like Professor Adler said, thank you so much for this presentation. It was very insightful. We do appreciate you joining us, Dr. McCutchen. Thank you.
Thank you for coming, and hope you learned something.
Great. Thank you so much. Have a great day, everyone.